Good afternoon, good morning, good evening, wherever you are. I'm very pleased to invite you to this, the first of the webinars that IPVS is going to give in 2021. These are supposed to be educational and informative and also a bit of fun. Now we have two fantastic speakers today. John Doorbar, who is a professor of virology in the University of Cambridge. He's a great friend of mine. In fact, I have the office next to him. And he is going to look at this question about latency from very much the virologist and by a scientific point of view. And we have Barbara Moscicki, who is a clinician, an epidemiologist, and a scientist, and she's going to look at latency very much from the clinical point of view. So perhaps we can start with John, who hopefully will now share his slides and hear about latency from his point of view.
All right. Okay, thank you very much, Margaret. Hopefully everyone can see my slides now. Yes, I was going to just describe a little bit about latency from the virus perspective and from the virus disease perspective. And I was going to start by just giving you a little bit of background to virus infections in general. And of course, comment that viruses can cause infections in a number of different ways. You're all very familiar with acute infections, such as those that might occur from corona or influenza virus. And in these type of situations, the virus causes disease. The disease is cleared by the immune system. The viral antigens are removed from the body. Waning immunity then follows, allowing a second infection at a later time, which may be less significant than the first infection because of the remnants of the previous immune system. Now, papillomaviruses are a little bit different. They are part of a group of viruses which cause chronic infections. And typically, human papillomavirus infections last not just for weeks, but often for weeks, months, or even years. And papillomaviruses are like many other viruses in causing chronic infections. And other examples include HIV, herpes viruses such as Epstein-Barr virus. And a common feature of viruses which cause chronic infections is that they often have a reservoir of infection. They have a cell type where they can persist in long term in the body without causing cell death of that cell they infect. And of course, for the papillomaviruses, the reservoir cell is the epithelial basal layer. These are epithelial cells which line the very lowest layers of the epithelium. And a typical characteristic of viruses which cause these type of reservoirs is the virus doesn't need to express very many genes. It avoids immune detection. And by these methods, the viral genome can persist in a particular cell type. And with papillomaviruses, virus production only occurs upon epithelial differentiation. It only starts when the reservoir cell divides and produces a daughter cell, which leaves the epithelial base layer. Now, we can see this if we look at a productive lesion caused by a low-risk papillomavirus, for instance. And here we've got a productive lesion showing the basal layer, showing where the basal reservoir cells are. And here we've stained in brown for markers of viral gene expression, in this case E6 and E7. And it's actually very, very hard to see viral gene expression in the basal layer because there's very little gene expression. There's also very little viral genomes there, only 50 or so copies, and the virus can persist here without being seen by the immune system. And this is central to our understanding of latency. Now, as these cells divide and produce cells which move towards the surface, you can see some of these cells stain brown because they're expressing viral gene products. And particular E6, E7 are essential for the virus to support viral genome amplification and eventually virus production. And the genes which are required for maintenance of the viral genome in the base layer, just to mention one of the papers we produced on this recently, are E2, probably small amounts of E6, or maybe little else. Now, this is a picture of a lesion, but of course we can show this diagrammatically like this to show the basal cell reservoir and show the sites where genome amplification and virus assembly occur. Now, chronic infection and these active infections are very important because it's deregulation of viral gene expression in this type of productively infected tissue, which starts off the whole process. It's deregulation, which is manifest eventually as neoplasia and eventually as cancers. But the whole situation is a little bit more complicated than this. And chronic infection is preceded by two other different types of states. So we've got immune control, which can occur when the lesions eventually do become cleared. And we've also got a situation which occurs before lesions actually appear. And for virologists, these are given many different names, sometimes the incubation phase or the lag phase. And when the lesion is cleared, the immune system can control the infection. The immune system clears the virus, clears the active infection, and immune control leads to a situation where HPV DNA can detect it without disease, or so it's thought. And the lag phase is another place where HPV DNA can be detected without disease. So let's take a look at these two states. These are often lumped together with a general term of latency. But I think latency is an often misused term. It's really, these two different states are quite different. And I don't think latency is a term we should use for these two. So let's stick to lag phase and immune control for the moment, and I'll show you a particular part of the immune control, which is latency, as the talk proceeds. So let's look at the lag phase. Now, it's said in the literature that this phase can last for weeks, months. Sometimes with vertical transmission, it's suggested it may even last for years, although I'm suspicious of that. Let's look at how it works. So if we look at the epithelium, the virus has to gain access to the epithelial basal layer. It infects one or more cells, and it imparts on these cells a growth advantage over its neighbors. And these cells start to expand in the basal layer. And this is probably helped by the wound healing response, and the virus-infected cells continue to expand until they reach a self-limited state, which is probably helped along by the mechanical pressure of the neighboring cells. And this reservoir infection, as it becomes suppressed and reaches a steady state level, these cells will then differentiate and complete the productive life cycle as shown here. Now, a point about the lag phase is that the duration of the lag phase seems to depend on the inoculating virus titer. And I'll show you one or two little bits of evidence that tells us this from our recent work. So if one infected cell starts off the disease, then it takes a long time for a lesion to appear. But if more and more cells start off the disease, if the inoculation is higher, disease appears quicker. I just want to show you one picture from a recent paper which show you how quickly disease occurs following inoculation of different titers of virus. And I just want to show you two here. When the virus titer is high, disease can occur in a few weeks. When the virus titer is low, well, at least in this model, it takes weeks and actually over 3 months before lesions start to appear. Now, I don't want to spend most of my time talking about the lag phase. I want to talk mainly about immune control. And the model here is that when disease is cleared, it's an immune response to the virus, and T cells recognize eventually the virus, and they come in and they control virus infection in the cells that they can see. But of course, the basal cells with the viral genome can't be seen by the immune system because viral gene expression is so low. So if we just think about viral latency, virus latency is one part of that immune control process. And I thought I would just stop a little bit and explain this to you. And I thought, well, it was easy enough just to pick a comment from Wikipedia because it's fairly accurate. Latency is a phase in certain viruses' life cycles in which, after initial infection, production of virus particles ceases. However, the viral genome is not eradicated. And this definition says the virus can be reactivated to produce productive infections later, and goes a little bit further than I would and says that the virus can stay in the host indefinitely. Another important point is it separates true viral latency from clinical latency. And the lag phase or the incubation period is one case of clinical latency. It's not quite the same as the state that's marked in red here. So if we just look at how virus infections occur, if we, following infection, viral gene expression gives rise to a papilloma or a flat wart, depending on the virus. And if it's a high-risk virus, well, deregulation of gene expression, followed by a failure of the immune system to clear, will in time lead to cancer. But of course, this is a rare outcome of infection. And what's more usual is the virus infection will persist. And the virus infection persists because of immune evasion capabilities of the virus. But eventually, in time, it's recognized by the immune system, leading to disease clearance. And I think the story I'm telling you here is disease clearance isn't the same as clearance of the virus. It's disease control. So now we can start to think about this in the context of what happens during a woman's lifetime, for instance. I just want to go over previous thinking and then come back to how our thinking may change if we take on the concept of virus latency. So as you know, with high-risk papillomavirus infections, affections occur following the onset of sexual activity. Eventually, for most people, immune recognition clears the disease. And if we see a new HPV detection later in life, this is often thought to be due to a new infection, either by the same HPV type or a different one. Now, this model, if we think about this model and think about what I've just said, we can also think about some of the epidemiological data. And there are many papers sort of highlighting the fact that this model doesn't easily fit well with what people observe. For instance, if we look at new infections during life, 12% of new infections occur in women who are currently at this time sexually inactive, which doesn't fit with a new HPV infection. 70% or so occur with women who are having sex with the same partner. Again, it's not easy to fit this into the idea of a new infection. And only 20% or so occur in women who report a new partner. So I think we can think of an alternative way of looking at this, and the immune regression leads to disease clearance. But clearance of disease doesn't mean clearance of infection. What it means is immune control of viral gene expression and immune surveillance, and this is common for many chronic infections. So if we come now back to our original idea, new infection can be lead to a new lesion, but also lesions may appear during changes in immune status, and occasionally the levels of HPV activity might rise above the level where they can be detected as a clinical threshold. So HPV latency, is it real and does it matter? Well, if we think of latency as an immune control, it's very important to understand how the virus is regulated. And if we look at this model, then we can anticipate that this will impact our interpretation of HPV DNA test results. So if we just now, are there some examples of the way papillomaviruses are controlled by the immune system? Well, I think yes. And we have to move here, not from the alpha papillomaviruses, but now to this other big group, the beta papillomaviruses. And these are a group of papillomaviruses which we are all infected with. They cover our whole body, and they're controlled by our immune system. So people, we don't know we have them. But these viruses are typically found in hair follicles, and eyebrow hairs are a rich source of this type of papillomavirus. And here we've got a hair follicle that's infected with a beta papillomavirus. And if we use an immunostain to show where L1 is, here it is. But this is an immunocompetent individual. And if we stain another marker here, E4, we can see E4 and L1 is produced. If we switch now to an immunodeficient individual, the level of viral gene expression is much higher. And the level of virus gene expression can actually spread, and we can find these lesions quite large. In fact, there's a basal cell carcinoma arising in the presence of an extensive region of viral expression here. And I think we get the idea that the balance between immune surveillance and HPV gene expression controls disease outcome. And if immunosurveillance is high, we see a situation like this. But as the immune system declines, the virus can get away with expressing more. Now, I just want to show you one or two more examples of this, because this is in people, but we can look in model systems of disease too. And one of these is the rabbit. These are experimental infections on the rabbit tongue produced by rabbit oral papillomavirus. And because they are experimental infections, we can follow them over time. So infection leads to the formation of disease. This is a papilloma. And these are in naive animals. So there's lots of gene expression and no immune surveillance. Over time, the immune system notices the virus, clears disease and we change the balance. And we end up a state where immune control controls viral gene expression. Now, because this is an animal model system, we can subsequently immunosuppress the animals and we can see what happens. And if we look at the way viral DNA copy number changes over time, of course, during lesion formation, it's high. It declines on immune regression. If we immunosuppress, it goes up again. So we get the idea of immune surveillance and viral gene expression being balanced. And this changing is the different immune control changes over the animal's lifetime. So if we now just come to what happens in people, I just want to quickly go over a study which we've just submitted for publication, where we looked at 100 hysterectomy samples from patients, trying to find out any samples where there was viral DNA present in the absence of disease. And from these 100 hysterectomies, we found 17 where there was viral DNA in apparently normal tissue. And a few of these were also positive in the smear. And I just want to show you the results when we started to use our RNA in situ detection probes. So these have normal pathology. These are not disease cases. If I just remind you about how the viral gene expression patterns look in an active productive infection, this time we're looking at E2, not E7. And we see a little bit in the base layer. This is a more sensitive method here. And we see elevation in gene expression in the upper layers. In these latent or inapparent infection cases, we find that the levels of E2 change, and they go from very, very low levels to patterns which are starting to resemble what we see in productive infections. And I think the idea is these represent immune-controlled infections in individuals who once probably had active infections. And the extreme of this depletion of immune response is that we start to see microlesions again. In one case, we saw a microlesion where the viral gene expression and E4 expression was starting to appear again. So in the last few minutes, I just want to finish up by showing you an animation of viral latency and immune control, which we prepared some years ago, which I think is still relevant. So if we look at this virus infection, the virus is hiding from the immune system because it has immune evasion strategies, but eventually Langerhans cells will see it. The Langerhans cells will pick up foreign antigens, and they will take these foreign antigens to the local lymph node. When they reach the local lymph node, the current thinking about immune regression is that the T cells become activated, and activated T cells are recruited to the site of infection. When these T cells reach the site of infection, some of them accumulate under the lesion and some of them enter the lesion. And I think the thinking here with the viral latency is that as the T cells arrive and start to produce cytokines to control the infection, viral gene expression subsides and the virus infection disappears almost as the T cells arrive. So we get a suppression of viral gene expression, we get immune surveillance, and this model really leads us to the possibility that over time, the number of infected cells may decline, but they're controlled by immune surveillance. And so it gives us the possibility that as the immune levels decline, perhaps in old age or in for other circumstances, the virus can start to get away with expressing more genes. And it's this balance between the immune cell noticing the virus and the virus expressing antigens, which keeps the immune cell active and is the basis of immune surveillance for many chronic infections. So if I can just conclude, summary of our current thinking, infection, T cells, memory T cells, suppression of viral gene expression is we think how the virus is controlled, and as immune status is modulated during life, the virus has a chance to produce virus particles now and again and to increase its copy number to levels that can be detected by clinical tests. And of course, if there's immune failure and this system doesn't kick in, then L-cell can progress to H-cell to cancer. And depending on the epithelial site, H-cell may occur more easily and cancer occur more frequently. So there are many different sites where the papillomavirus can infect, but critical for infection is this transformation zone region. And I think in the future, we want to look at the different cancer paths of this site and also consider how the immune system controls infection at these sites. I realize I'm big risk of running over time, so I'll stop sharing at that point and return you, hopefully, to Margaret. Thank you very much.
Thank you very much, John. That was absolutely splendid. And because we have to keep very tightly to time, we'll answer all the questions in the Q&A. So I'm now going to ask Barbara Moscicki to give you a talk that looks at the clinical aspects of latency. And because she's a Californian, it's what does it mean existentially? Thank you, Barbara.
So I'm going to be talking more about latency in what we call epidemiology or epi terms. So usually that means, yes, detect it, then you don't detect it, and then you detect it again. So the big question is, when are we really convinced, a negative test means actually not any viral infection? So this is a little bit of data showing intermittent patterns of what we saw for HPV 16 in our San Francisco cohort study that went over actually up to 25 years. So we have long-term data where women were tested every 4 to 6 months. And you can see like the top pattern right here, this woman, very intermittent, but if we had done only 2 tests on her, we would have thought, in fact, that woman is negative, and then she had long periods of being positive. So the question is, when we see 2 negatives, are we more convinced that, in fact, maybe these are new infections that are occurring, or are we asking for 3 negative tests? So a lot of this is a little more confusing on how we define negativity. So John just went over a very eloquent discussion on dormant or chronic low-level infections, but certainly other possibilities are sampling or amplification error in the test itself. And certainly what I'll be focusing on is reinfection from a partner or self or autoinoculation. So this is a study from our cohort where we identified women who had cleared HPV 16, and then we saw a second detection of it over time. And almost, you can see here, around 20% over 9 years, we were able to detect 16 again. And then we looked at clearance of those and then recurrence. And I'm going to go over this a little bit more detail. So the solid line is prevalent and the dashed line is incident. And what you can see is the first infection and the second infection were cleared both rapidly and just about the same time, suggesting that, I know these scales are a little bit different, but the clearance at 2 years was almost identical in both groups, suggesting perhaps that these were both kind of new infections. When we looked at factors associated with clearance, we see that it looks like condom use and any sexual contact, any sexual contact versus none, slowed clearance, condom use, hastened clearance. So once again, somehow protecting yourself from your own partner actually helped from helped in the clearance of that virus. So then we look a little more careful at the second detection and third detection. This I thought was really interesting because if we look again, the prevalent versus incidence, pretty tight, no really difference there. But by the time we saw the third one, the prevalent infections who are initially prevalent, which much more likely to see that individual with 16 again for a third time. Whereas of the incident ones, you can see it's a straight line we didn't really detect. So the bottom line is, I think some of these patterns, especially if they come in with a prevalent infection, these women are much more likely to be these intermittent detectors over time. So what about, we looked at those factors that are associated with redetection among those women who cleared, who I first showed you, and very likely associated with sexual behavior. That is, they had more than one partner in the past month, they had a sexually transmitted infection, and they also reported a new partner. So all of these were associated with redetection. Then we kind of did a little twist on how we looked at this, and we also had serology on a large portion of them. So then we looked by seropositive status again. So we identified women who were both seropositive, seronegative, and we looked at acquisition and then clearance and then reacquisition. So in theory, again, looking at this a little closer, what we can see is that those women who are seronegative in the dashed line are more likely to acquire 16 over time than a seropositive group. And we know that there might be some protection with natural history. We also saw this to be true, but even more so perhaps in the third detection. When we then looked at clearance, however, they were pretty tight about whether, there's small numbers over here, but both cleared equally and very strong and over time. So then we looked at some of those factors for the acquisition, again, the seronegative, and I'll show you the seropositive. So if you look among the seronegative, looks like it was a lot of sexual behavior, new sex partner, the STI, having more than one partner, as well as alcohol use and lack of condom use. But when we looked at seronegative, we did see a slight difference. I'm sorry, the seropositive group, slightly different, in that cigarette smoking was associated, which also has been associated with high-risk sexual behaviors, and also reporting more than one partner over time. And what we see also, however, is we identified, somewhat surprisingly, the use of medroxyprogesterone or Depo-Provera as birth control was associated with this. And we looked at the clearance group, and of interest, having also use of medroxyprogesterone was associated with slower or lack of clearance. So actually we thought, going back to this slide, that it's not really the use of medroxyprogesterone that we see for acquisition. It ends up in this population that they're not clearing this virus actually quite well. So they actually end up being a pattern of probably one of those very intermittent or long-term individuals who are carrying that chronic infection. We also took the opportunity in our study, we were looking at cell-mediated immune responses that John was referring to, using the serological response using PBMCs, and we did interferon gamma or granzyme B ELISpot, and we looked at their responses to HPV 16, E6, and E7. Of what we noted, we had 21 women in the cohort that we identified a positive cell-mediated immune response, and what we found is 15 cleared with no HPV detected ever, and there was basically a range from 7 to 25 years. We also looked at 3 women who had a shorter period of intermittent HPV 16 detection, but they eventually cleared it and had an average of 19 years of clearance, that we were not once able to detect HPV 16 again. And 3 women, however, did have intermittent detection over the time, again, ranging anywhere from 7 to 25 years. And 2 of these women went on to develop CIN2-3. So this kind of suggests that we do have these women who truly clear, but these other women are carrying this very chronic low-level infection, which is intermittently detected. So what other evidence do we have? This is a paper from Tatya et al., and she looked at HPV, the risk of HPV with new sexual partners in a relatively large age group. And what you can see here is that whether it was initial infection or reinfection, they were all highly associated with a new sexual partner. This was a study by Thielen et al., where they looked among HIV-infected, sexually absent women. Though these women did acquire it, it was only acquisition, only abstinent for 6 months. But what was more important is that we would have thought if they were immunosuppressed, we actually would have saw some statistical difference between those with less than 200 versus greater than 500. And none of these were statistically significant. So this paper also was able to look at risk factors in women aged 35 to 60 years. And this looked at all women, not surprisingly. Once again, new partner was a risk factor, but lifetime partners also were. They did a separate sub-study and showed that the new partner, among those that were sexually absent, so partners were no longer, but lifetime partners continued. And I think it's really difficult to say just because you have a lifetime partners, so that's why you're going to get latency, because we know that it's very complicated, the transmission rates between partners and also sexual networks, and as I'll go through, also autoinoculation. So this paper also looked among those that were in the control arm of the Vivian trials. That was not a long trial, but once again, if you were seropositive or seronegative, number of sex partners during the last year had elevated hazard ratios. So this last paper I'll just refer to looked at young women, but they had a average time of 6.4 years. What you can see, a lot of sexual behavior was associated with acquisition, or I said redetection, and that included the number of sex partners, having a new sex partner, unprotected sex, and as well as then we saw oral contraceptive use, which often is correlated with condom use and sexual activity. So what's another thing? We say, oh, we see all this HPV in older women, but we also know that actually syphilis and chlamydia are actually on the rise in this older age group. So sex does not end after 65, and that's important to remember. And that includes the partner. And monogamy is not always see-through. So what about other sources? We love to blame our partner. This was a study that we did looking at transmission. And so we brought in couples who said they were monogamous for at least 3 months. And we tested them at 0.1. And then we asked them to basically have sex and come in within 48 hours. And then we asked them not to have sex, that third visit here. And so what you can see is that just recently having sex, there was a lot of sharing that we went on, but also that if they were positive, one partner was positive for an infection in the genital area, the other partner had it in their anus. And we see that true with the hand, less likely from the oral. So certainly the partner as well as the anus and the hand of the partner could be a source of infection. We looked at just the transmission rate per 100-person months, and what you can see, just over a 6-week period of time, both men and women had, transmission events, showing how easily it is to transmit between these monogamous partners. And also what we found is they had multiple infection, and we could only explain around 15% of those infections. That is, the partner had it and we looked elsewhere, the hand, the oral, anywhere else in the genital, and we could not find it around 85% of the time, showing that there probably are different places where the virus can exist that we don't know and then be later transmitted. As I had mentioned, sexual networks can be very complicated and often actually very close and tight-knit, so that without knowing, other partners are often having sex with others, whether it's short-term or long-term. This is a paper out of Goodman, once again showing that the anus may be a reservoir because they had a relative risk of 8 or almost 9 of a risk of acquiring a cervical infection after having an anal HPV infection of that type. So what about the vagina itself? Well, there's been, correlations anywhere from 0.4 to 0.9 have been reported when you compare self-sampling vaginal swabs to cervical collection by a clinician. So this was a study that we did where I was mentioning to you that we had the women coming in every 4 to 6 months. And we actually sent them a sample in between those visits to do a self-collected vaginal sample. And this just shows you right here that we saw very similar clearance rates, whether this was from the vagina or collected from the cervix. However, we did show that there were some similarities on what was the fastest, 6, and what was the slowest was HPV 62, but other types were clearing very differently within the vaginal and the cervical samples, suggesting that because you clear it in the cervix, it still may be hanging out in the vagina and could be transmitted again. So we looked a little bit more careful between women who then had 2 positive tests for the same type from vaginal samples, but were negative for the cervical cancer. And then vice versa, they were positive for the cervix, but negative. And so what we saw here is that about 1/3 to 40%, in fact, were discrepant. So that somehow we were measuring things from the vagina, but not the cervix, showing that the vagina could remain actually an important reservoir for detection later. So going down to the clinical relevance of this. So does having a past history of, an abnormal test make a difference of screening a test today? That is it a prevalent or is it an incident? So, dear doctor, my previous screening was negative. Now my test is positive. What does that mean for me? So I think we always know one of the discussion points is the easy answer is, oh, it's just likely a reactivated latent infection. So the hard answer is, do you think this came from your partner? But perhaps the right question really is that HPV testing is a screen for cervical cancer. So let's talk about your risk for cancer. This study that came out of Costa Rica showed that if you had an incident, no matter what age you are, all the way from the younger ones, 26 to 33, up to 42 and more, these infections all cleared almost identical. It was only those prevalent infections in the older women that were much slower to clear, suggesting these women already entered with a chronic infection. So this came from also the Kaiser Permanente data that was actually used for the new risk management guidelines. And what they showed was if you had an HPV positive test, that you had a cumulative risk of around 4% if that test was a prevalent test. But if you had a known test that was HPV negative, it fell in half to like 2%, which was no longer meeting our even colposcopy threshold because the risk of CIN3 was considered low for the risk itself. So this also came from the Kaiser Permanente data where they looked at rounds of co-testing. So once again, once you had one round of co-testing, the 5-year cumulative incident was only around 0.6. If you had already 2, it drastically fell. So clinically, once you become negative and then you are positive, those results suggest a very low risk for that woman. So in summary, I think most HPV infections truly do clear. And with long intervals of negative tests, these new incident infections likely reflect infections from partners or autoinoculation. And these redetected infections rapidly, these redetected infections, I think, immediately will clear and are at low risk. But what about reactivation? I do think that that can happen, but I think these actually reflect chronic ongoing infections with very periods of low replication, and these women remain at risk for cancer development. So this is my existential map of latency. You are here for no apparent reason. Thank you.
Thank you very much. That was terrific, Barbara. Now, this has excited 29 questions, so I'm going to exercise my role as moderator and select them. And a sort of recurring question, which I think both of you can answer, is, would you expect HPV vaccination delivered after the initial HPV infection to protect against reactivation or to assist with clearance of reactivated infections? So any thoughts?
Well, shall I just, I guess what we're thinking about, so I think when we talk about latency, I'm saying it's immune control, and it's an extreme version of immune control. And the immune control on the virus has to be to the viral early proteins, which control disease. So I would think it would be in a T cell response to E7, E6, E2, and it might be different in different people. And I wouldn't strongly be thinking that an antibody to L1 would be so important. That's my naive view for now. Yeah.
Barbara.
So I absolutely agree with John that I did not think that it would do this. However, we wanted to look at redetection in that subgroup of our women who still were age eligible to get the vaccine during our cohort study. And I wanted to look at the local cytokine response in women who had like 16 or 18 before they got their immunization, and then look at if we saw redetection. And unfortunately, we could not find any of those women. There were virtually none who had gotten vaccinated who we saw 16 show up again. I don't know why. It could have been, small numbers, and we didn't that. So I was really surprised to see how protected these women looked, even though we saw 16 show up, before they even got vaccinated.
Well, there's a possible answer, which is that, and it goes back to John's point, that you build up the number of infected cells, probably from autoinoculation. And if you have antibody to L1, you will actually block that autoinoculation. And that's really been raised as a possibility for the use of antibody against L1 in RRP. So there are reports from respectable laboratories, respectable groups, of using the vaccines, the target 6 and 11, in patients with RRP and getting a positive outcome in a proportion. And I think the idea that you reactivate and you get a very little tiny column of cells which shed L virus, and that can be neutralized by antibody that's in the vaginal fluids, for example, or in the larynx and trachea. But that's a speculation rather than a fact. But there's no question that one of the most commonly asked questions on this webinar is, what is the impact of vaccination on infection, on latent infection? Do we know?
You mean when a latent infection is already present?
Yes.
When an immune controlled. So I would like to use the immune control more than latency, because latency is a subset of that. I don't think there will be. A lot of it will be down to autoinoculation. So you're just increasing your antibodies primarily, and maybe some T cell responses to L1. So I'm not thinking that L1 is the key to changing latency. I'm thinking it's
No, I'm not arguing that it is. I was simply pointing out that if you vaccinate against L1, you do have the possibility of protecting against autoinoculation.
Yes.
Yeah. Auto and as well as partner. I think, like I said, the partner is carrying a lot that is not always detected. So as long as that individual doesn't have that partner's HPV, they're always going to be at risk of then acquiring it at some point in time. So I think that is what is also going to be protected, both, auto inoculation and, from the partner itself.
And we have a question from Connie Trimble, targeted to John. Yeah. What is the role of tissue-resident memory immune cells?
Well, I was thinking I could bounce that one back to you, Margaret, actually. I mean, I think, I mean, for my thinking, I haven't massively got there. I just put T cells with a little T on, and that I know that's ridiculously simplistic, and I can get away with that because it's a 15-minute talk, and there isn't space to say anymore. I think the way I see, immune surveillance is the tissue-specific memory T cells is extremely important, and they'll constantly be bumping into viral antigens when the virus produces them. And they'll be stimulated locally to produce cytokines. This is, again, a naive virologist point of view, and that'll tend to shut off viral gene expression. So it'll be almost continuous and imperceptible, but it won't always require a lymph node involvement. They'll just be there. There'll be memory T cells in the epithelium and the viral, and so you've got this constant tweaking of the immune system to keep it active for a long time. So
yeah.
never disappear from the
I mean, I think there definitely would be a role for tissue-resident memory cells, because the functional CD8 resident memory T cells are actually intra-epithelial rather than not in the stroma, and their role would be to get kicked back into action as soon as there was any viral reactivation. So I would say that there was a role for it. But why that role is suppressed when, as you go through a woman's life, it must be a very local immune control. Another question is to Barbara. If lifetime partners aren't a good proxy for trying to better understand potential for latency, what would you recommend? What do you think is a good proxy?
Oh, if lifetime partners. So, the thing, studying sexual behavior is very difficult. Number one,
because they all lie.
Well, there's the issue of, honesty. And we know that it's not terribly uncommon that even in what we consider, monogamous relationships. I didn't show a slide that showed if you had risky behavior, you had 10-fold chance of looking still risky 5 years later. And this was true both for men and for women. So the example is that if, even though a partner may have 5 in some long time ago, and now they're in a relationship, but their partner had 20 different partners, and now they're in a relationship. So even though the woman says, I haven't had sex with anybody else, she's got a partner who had certainly a history of high-risk sexual behavior. So, whether that is another occasional extension of their sexual network is the difficult part. So I think, lifetime sexual partners is kind of an interesting thing that happens sometime. And certainly every time you have more partners, we know you're more likely to get cervical cancer. So that would almost be saying, well, the more number of partners you have, the more likely you are just to, have chronic infection that's going to go on to cancer. And I think that's kind of, I think looking at lifetime partners is a difficult one. And I'm not sure there is any proxy if you're saying that that's the risk that the woman has multiple infection and latency. I don't see how lifetime partners can be really used to predict a woman who's going to have a latent infection or immune control versus non-immune control.
Can I make just one comment? Because I think the whole thing is quite complicated, because when we did our hysterectomy study, we found that the more partners they have, the less chance of latency. And I almost thought it was something, the more exposure to an antigen, the more likely their immune system is getting more stimulated. So there may be some counterintuitive things going on there, which I don't think we can delve into because there aren't enough studies in this area.
Yeah.
There's a question from Sylvia de San Jose, and she, first of all, she says, great talks. Barbara, is there any effort being made to do genotype sequencing? In other words, to look at whether these other, you get a positive result, then you get a negative, then another positive later. Is it actually the same virus, or is it maybe the same type, but it may.
So, we in fact published a really tiny study and we tried to look at this, and unfortunately we did not have great success doing the sequencing on the second one on paired samples. But of the ones that we did, we had about 50% had the same, HPV type, and 50% were different. So, said that, even if they're the same type, it doesn't prove that it wasn't autoinoculation, and it doesn't prove that it didn't come from the partner. Mm-hmm. Especially that if, the partner then has it, you clear it, they give it back to you. So I'm not even sure when we're saying it's the same sequence, that absolutely means that that's not autoinoculation or partner inoculation.
Yeah, I think what you're saying is that probably these studies are almost impossible. Betty Steinberg, who knows more about latency than any of us, I think, has made a couple of comments. And she's pointed out that if reinfection from partners or autoinoculation is a major cause of recurrent infection, then the vaccine should be highly effective, which is what you showed, Barbara, in one of your studies. And she also comments on my comment about RRP. The data on RRP is weak. It's anecdotal. The vaccine therapy studies weren't placebo-controlled, and the placebo effect might be very important in immune function. Now we've got a huge number of questions here, and I'm trying to find out. A question about innate immunity. What's the role of innate immunity in control and intermittent infection? You've done quite a lot on this, Barbara.
Yeah, so we published that paper again using the cohort. So we identified women who were negative, they acquired HPV-16, and then they cleared, versus a population where they acquired it, but they persisted at the next visit. And what we found is, the toll-like receptor, specifically 7, 8, and 9, became super expressed right prior to clearance. Whereas we saw almost a suppression of the TLRs in those women that had persistent infection. So I think innate is certainly responsible for, this kind of rapid clearance. Of interest, we then look further at women who cleared after, several months to years of being positive. And we showed once again that the TLRs were very important, but were very much synced with the cell-mediated immune response. So the cascade of TLRs, right, can not only trigger innate, but TLRs are also important for the development of adaptive immune responses. And we found those highly correlated in our group as well.
There have been several questions saying that we've been focusing on the vagina and cervix, and that I think is because that's where most of the data's come from. But have we any evidence that these concepts apply generally to other sites of infection, such as the anus or the tonsil?
So we did just recently publish, the anal natural history, and it pretty much looked like the cervix, that most of these infections clear. And, kind of anal sex, anal touching, finger sex was actually slowed clearance. And again, not perhaps too surprising. So we don't have as much, I think, data, but the one slide I showed you from the Goodman article basically showed that having an anal infection preceded a cervical. What I didn't show you is that a cervical infection also preceded detection of HPV from the anus. I mean, they're only that far apart, Yeah,
I think, this is an infection of squamous epithelium. And a lot of it is about the biology of the keratinocyte. And as John, I think, was alluding to.
I think that, you know, the whole story about beta papillomaviruses, which you're infected with before the age of 5, and which you're HPV positive in swabs for many, many decades afterwards, really indicates that you can call them subclinical infections, but subclinical papilloma infections seem to exist, and we can apply that to mouthwashes. You know, you 5% positive in mouthwashes without apparent disease, and when we take punch biopsies, we can find HPV and productive infections in salivary glands, and we published some of that with Keith Hunter from Sheffield. So these viruses are existing in us in many different places, and I think they're immune controlled, and the immune control is just latency is one end of the spectrum, and chronic active infection is another end of the spectrum. And in between, there's a whole host of things.
I'd like to go back to Barbara and ask a question which is really quite important, because lots of people looking at this webinar are clinicians, and they would like an opinion from a practicing clinician. Is a latency really important? Does it matter? Does it matter clinically?
I don't think it does. So again, because the data that looked at risk of CIN3, right? That's the clinical data we want to know, is extremely low if you have a documented incident HPV infection, at least the risk over the next 5 years. So if you are negative and then you become positive, whether maybe that's latent or whether that is autoinoculation or whether that is a new partner infection, the risk over the next 5 years is quite low. So what's important, though, is bringing that woman back in. And if she continues to be, you know, persistently positive tests, that tells you even more information that she does become at high risk. So, you know, ultimately, I don't think whether that is latent or reactivation. I do believe that these chronic low-level infections, these women are at much higher risk. So once they're kind of positive, those women really, and persistently positive, they really need close surveillance. Less so if they're incident positive.
I think we haven't addressed another group of individuals who really are at risk clinically if latency is real. That's people who are becoming immunosuppressed post-infection, for example, transplants, various sorts of autoimmune diseases which come on later in life. And so I think we have to look at that in that context. I don't know what either of the speakers thinks about that comment.
Well, I would say that, you know, chronic viral infections are always a problem in immunosuppressed, and they'll always come back. And we've got many, you know, Kaposi sarcoma and EBV, and all of them are a problem. So HPV is the same. I think when you're immunosuppressed, you have the chance of the reservoir increasing in size.
Yeah.
The whole situation deteriorating.
Yeah, absolutely. Well, I think that was a terrific pair of talks, and I can only say thank you very much. And thank you to all the people who dialed in, and I hope you enjoyed it as much as I did.